Swayam Prabha. Digital India. Educated India.
Hi students, I am Dr. Anandrani, Professor, Department of Anatomy, Sri Ramachandra Medical College, Srihar, Chennai. Today's topics for discussion are histology of cornea, sclerocorneal junction and retina. Objectives are here. At the end of the session, the students should be able to enumerate the layers of cornea, describe the microscopic features of cornea, describe the histology of sclerocorneal junction, list the layers of cornea, describe the histological features of retina. Regarding the applied histology, at the end of the session, histological basis of the following conditions will be discussed: corneal opacities, glaucoma, retinopathy and retinal detachment. Eyeball occupies the anterior third of the orbital cavity formed by segments of two spheres. Anterior 1/6 forms the cornea, representing a smaller sphere. Posterior 5/6 forms the sclera, representing larger sphere. Anterior and posterior poles of the eye are the central points of corneal and scleral curvatures. Optical axis join these poles. In this diagram, you can see these two segments. This is the anterior segment, which is formed by the cornea, and this is the posterior segment, which is formed by the sclera. This forms part of a smaller sphere and this forms part of the larger sphere. Eyeball encloses the refractive media. The wall of the eyeball is composed of three layers. The outer tunic is fibrous and consists of sclera and the cornea. Here you are seeing the fibrous tunic or outer tunic, which is composed of two parts. This is the sclera and the cornea in front. The intermediate tunic is pigmented and vascular, includes choroid, ciliary body and iris from posterior to anterior. Here is the vascular tunic, which is intermediate, and here are the parts: choroid, ciliary body and iris. The inner tunic is nervous and is formed by the retina. This is the inner tunic, this is the retina, and this is having two portions: the pigmented layer close to the choroid and the neural layer inside. Coming to the cornea, it becomes continuous posteriorly with the sclera at the limbus, which is otherwise called as sclerocorneal junction. It is transparent and is the colorless part of the fibrous layer. The thickness of the cornea varies: 1 mm at the periphery and 0.5 mm in the center. Maximum refraction of light takes place from here. Refractive power of the cornea is much greater than that of the lens. Cornea is avascular and is devoid of lymphatics. It is nourished by diffusion from adjacent capillaries at the ends, aqueous humor from the internal surface, and central parts from the dissolved oxygen in the tear. You are seeing the cornea here, which forms the anterior part, and the sclera is behind, and you can see the cornea as the part of the fibrous layer of the eyeball. This slide shows the microscopic appearance of the cornea. The section is stained with hematoxylin and eosin and is seen under low power magnification. You can see all the layers of the cornea here, starting from anterior to posterior. Anterior most is the anterior epithelium, stratified squamous non-keratinized epithelium. The next layer is the anterior limiting membrane or Bowman's membrane. The third layer is the substantia propria, which forms the major bulk of the cornea. The next one is the posterior limiting membrane, that is Descemet's membrane. The last layer is the posterior epithelium or simple squamous endothelium. First is the anterior epithelium or corneal epithelium, made up of stratified squamous non-keratinized epithelium. 5 to 6 layers are present. Superficial cells have microvilli. These microvilli help to retain a thin film of fluid on the surface of the cornea to keep it moist always. This particular layer is having remarkable capacity of regeneration. At the limbus, 10 to 15 layers are there and becomes continuous with the bulbar conjunctiva. You are seeing the stratified squamous non-keratinized epithelium here. Anterior limiting membrane or Bowman's membrane, it is homogeneous and a cellular layer lying beneath the corneal epithelium, made up of compactly packed collagen fibrils and covers the substantia propria. This layer gives stability to the cornea. Here is the Bowman's membrane, which is next to the anterior epithelium of the cornea. Substantia propria, it is composed of 200 to 250 superimposed flattened lamellae. Bundles of collagen fibrils are present in the lamellae. These collagen fibrils run parallel to each other and to the surface of the cornea. Collagen fibrils are of uniform size and are embedded in chondroitin sulfate and keratosulfate. Flattened fibroblast cells called as keratocytes are seen in between the layers. In the microscopic view, you can see the substantia propria and you can see the collagen fibers. In between, you can see the nuclei which belong to the fibroblast cells. Posterior limiting membrane or Descemet's membrane, it is homogeneous and acellular, made up of compactly packed collagen fibrils. It breaks into trabecular meshwork at the limbus, that is at the sclerocorneal junction. Here is the position of Descemet's membrane. Endothelium, posterior epithelium, single layer of cells you can see here. Squamous or cuboidal cells form a covering over the cornea. At the limbus, it becomes continuous with the endothelium lining the trabecular meshwork formed by the splitting of Descemet's membrane. Here is the simple squamous epithelium, which is otherwise called as the posterior epithelium of the cornea. Now, coming to the vessels and nerves of the cornea, cornea is avascular. Allogenic transplantations of the cornea without any immunological rejection is mainly due to this factor. Cornea has rich sensory nerve supply derived from ophthalmic nerve via long ciliary nerves. The ciliary nerves form 4 successive plexuses: annular plexus at the periphery of cornea, proprial plexus in the substantia propria, beneath the corneal epithelium subepithelial plexus, intraepithelial plexus. Free nerve terminals penetrate the corneal epithelium and they form intraepithelial plexus. Transparency of the cornea is maintained by many factors. This property may be due to smoothness of epithelium, absence of blood vessels, uniform organization of collagen fibrils of substantia propria, thin collagen fibers compared to the other connective tissues, proteoglycans by regulating the diameter and spacing of collagen fibrils, epithelium and endothelium by serving as mechanical barriers to fluid diffusion and by creating a gradient that allows osmotic transport of water out of the stroma. Now coming to the next topic, sclerocorneal junction, which is otherwise called as limbus. You are seeing the microscopic appearance of the sclerocorneal junction. The section is stained with hematoxylin and eosin and we are seeing under low power magnification. We can identify the cornea here and the sclera here, and this is the position of the sclerocorneal junction. The transparent cornea becomes continuous with the opaque sclera at this point. It is marked by a shallow depression externally. A canal lined by endothelium is present here, canal of Schlemm, that is scleral venous sinus. This canal helps in the drainage of aqueous humor as it connects the anterior chamber through the foramina fontana with the anterior ciliary veins externally. In this microscopic picture, we can identify the canal here. This is a scleral venous sinus. we are seeing the boundaries of the sinus venosus sclerae. Sinus venosus sclerae is bounded on outer wall by a sulcus in the sclera. On the inner side, anteriorly, a trabecular tissue is bounding the canal, which is formed by the Descemet's membrane of the cornea. Here is the cornea and here is the sclera. Here you are seeing the canal. Posteriorly, it is bounded by a scleral spur, which is a triangular projection from the sclera. Anterior surface of the scleral spur gives attachment to the trabecular tissue and posterior surface provides origin to the ciliaris muscle. This slide shows the function and significance of the sinus. The sinus collects the aqueous humor from the anterior chamber, drains the aqueous to the anterior ciliary veins through the aqueous veins. If the drainage is blocked, intraocular pressure rises, resulting in a condition known as glaucoma. In venous congestion, the canal may be filled with blood. Normally, there is no blood in the sinus. This is the position of the sinus venosus sclerae, and here is the cornea and here is the sclera. This is the sclerocorneal junction or limbus. We are going to see the applied histology of cornea and sclerocorneal junction. The term corneal opacity means loss of transparency of cornea. Corneal opacity can be of two types, congenital and acquired. The congenital type of corneal opacity is usually associated with other ocular abnormalities. The acquired type is mainly due to penetrating ocular injuries which are resulting in corneal lacerations. The post-operative scar formation in these cases will lead on to corneal opacities. Glaucoma is a clinical condition where there is an increase in the intraocular pressure. The scleral venous sinus, which is seen at the limbus, is an endothelium-lined channel resembling a lymphatic channel. This channel helps in the drainage of aqueous humor from the anterior chamber. So, whenever there is a blockage in this channel, it can lead on to an increase in the intraocular pressure. We have seen the microscopic anatomy of cornea and sclerocorneal junction. Now, I am going to summarize these two topics. Here you are seeing the microanatomy of cornea, the microscopic appearance of the section of the cornea, which shows all the layers of the cornea. Starting from the anterior part, it is a stratified squamous non-keratinized epithelium. Next to that is the Bowman's membrane, then the substantia propria. Next to that is the Descemet's membrane and the posterior epithelium, that is a simple squamous endothelium of the cornea. This is the microscopic appearance of the sclerocorneal junction, and we can identify the sclera and the cornea. This is the sclera cornea, and here is the canal, sinus venosus sclerae or canal of Schlumpf, which is lined by endothelium. Now we are going to see the next topic, that is the retina, starting with this slide which shows the general features of retina. It is the inner coat of the eyeball which is photosensitive. In fresh specimen, it is pinkish red in color, soft and translucent. Two parts are there. The posterior 3/4 is photosensitive, extends from the optic disc to the ciliary body. Anterior 1/4 is non-sensitive to light and it lines ciliary body and iris. That is pars ciliaris retina and pars iridis retina. As a double layered epithelium, it lines the ciliary body and iris. The posterior part is called as the optic part of the retina and extends up to the ora serrata, that is crenated margin at the periphery of the ciliary body. You are seeing the ora serrata here. This is the ciliary body and this is the retina. This extends up to this part, that is the ora serrata. Here we are seeing some more features of the retina. The visual axis passes through the depression in the retina called as the fovea centralis. At this part, the retina is thin and few layers are absent here. The rods are absent, the visual acuity is maximum and the blood vessels are absent. The fovea is surrounded by an oval yellow pigmented area called as macula lutea. You can see the macula in this picture. The optic disc, which is otherwise called as the optic papilla, is about 4 millimeter medial to the fovea. Round pale pink area it is. The optic nerve's exit is through this point. It is pierced by central vessels of the retina. Since the photoreceptors are absent here, it is considered as a blind point. We are going to learn the microanatomy of retina, and here is the diagrammatic representation of the microscopic picture of the retina. Retina is photosensitive and has two layers: pigment epithelium, that is a non-nervous part, and the nervous layer. In this diagrammatic representation, you can see the pigmented epithelium here, and the remaining part is the nervous layer of the retina. The retina lies between the choroid externally and the hyaloid membrane of the vitreous body internally. Anteriorly, it terminates at the ora serrata, where the non-nervous part of the retina in the form of a bilaminar epithelium extends to line the deep surfaces of ciliary body and iris. In this diagrammatic representation, you can see the layers of the retina. The pigmented epithelium, that is the outermost layer, which is close to the choroid. Next to the pigmented epithelium, outer and inner segments of rods and cones, then external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic layer, nerve fibers layer, and finally internal limiting membrane. You can see the important neuronal cells of the retina also in this diagrammatic representation. The ganglionic cells, amacrine cells, bipolar cells, rods and cones, all these are the neuronal cells in the retina. Along with that, we are seeing one another cell here, that is the Muller's cell. This is the neuroglial cell which is present in the retina, that is a supporting cell which is present among the neuronal cells. You can see the cell body of the Muller's cell here, and the processes are extending throughout the retina. We are going to see all these layers in detail. Now you are seeing the microscopic appearance of the retina. Here the slide is stained with hematoxylin and eosin and viewed under the low power magnification. We are seeing all the layers starting from the pigmented epithelium, layer of rods and cones, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglion cell layer, nerve fiber layer, and inner limiting membrane. Coming to the first layer, which is the outermost layer, pigment epithelium, it is formed by a layer of cuboidal cells firmly attached to the choroid. Epithelial cells contain basal nuclei, abundant mitochondria, plenty of melanin granules, lysosomes, and phagosomes. The basal surface shows infoldings of the plasma membrane. Apical surface shows microvilli. Functions: absorption of light and preventing back reflection once the photoreceptors are stimulated. So here we are seeing the pigmented epithelium, and this is close to the choroid. This slide shows some more functions of the pigmented epithelium. Apart from the absorption of light and preventing back reflection, there are some other functions for the pigmented epithelium. Phagocytosis of parts of rods and cones that are discarded form an effective blood-retinal barrier between the retina and the blood vessels of choroid by their tight junctions. This gives a special ionic environment to the retina. The barrier prevents the entry of immunologically competent lymphocytes within the retina. This is the diagrammatic representation of this layer, that is the pigmented epithelium you are seeing here, and the remaining part of the retina will be called as the nervous part of the retina. We are going to see the microanatomy of photoreceptors, rods and cones. Here you are seeing the diagrammatic representation of a rod cell. 120 million in each eye, distributed around the periphery, absent in the fovea centralis, sensitive to light of low intensity, that is dim light vision, not for color discrimination. You can see the outer segment here, which is thin, long and cylindrical. And in the opposite end, you can see the rod spherule, which is knob-like. You can see the membranous disc, which is present in the outer segment here. There are many, and these will contain photopigment called as rhodopsin. Tips of the rods are constantly shed and phagocytosed by the pigment epithelium. Coming to the cones, 6 million in each eye, concentrated in the center, present in large number in the fovea, sensitive to light of high intensity, bright light vision. These are for color discrimination. Here you can see the outer segment, which is thick, short and conical. You can see the membranous disc, which is present here. There are many, and these contain a photopigment called as iodopsin. In the opposite end, you can see the cone pedicle, and here is the cone fiber, and cell body is present here. Coming to the layer of rods and cones, the outer segments of rods and cones form this layer. Membranous discs are present here. These contain photopigments. Outer segments of rods are cylindrical and those of cones are cone-shaped. Outer segments of rods contain rhodopsin, that is the visual purple, which senses low level of illumination and black and white colors. Outer segments of cones contain iodopsin, which senses bright light and colors. In the microscopic appearance, this is the view of the layer of rods and cones. This is the pigmented epithelium. Next to that, you can see the layer of rods and cones. The next layer is external limiting membrane. In fact, it is not a membrane, but junctional complexes formed between the retinoglial cells, that is the Muller's cell, and the inner segments of the photoreceptors will form this layer. This is the third layer, that is the external limiting membrane. Next is the outer nuclear layer, consists of cell bodies of rods and cones and their nuclei. In the microscopic examination, this layer will appear like this. This is the outer nuclear layer. Pale oval nuclei of cones lie nearer to the external limiting membrane, while the dark round nuclei of rods are deep to the external limiting membrane. Next is the outer plexiform layer. This is made up of synapses between the axons, that is the fibers of rods and cones cells, with the dendrites of bipolar cells and the processes of horizontal cells. The rods are polysynaptic with the bipolar cells, that is group of rods synapse with a bipolar cell. The cones are monosynaptic. One cone will synapse with a single bipolar cell. Now we are going to see the inner nuclear layer, which is formed mainly by the cell bodies of bipolar neurons. Cell bodies of horizontal cells, amacrine cells and Muller's cells are also seen in this layer. There are two types of bipolar cells, diffuse and monosynaptic. The diffuse bipolar cells have synapses with more than one photoreceptors. The monosynaptic bipolar cells will synapse with only one photoreceptors, mainly cones. The bipolar cells are the first order neurons in the visual pathway. Amacrine and horizontal neurons are association neurons. In this diagrammatic representation, you are seeing the sixth layer, that is the inner nuclear layer here, where you can see the neurons of bipolar neurons, Muller cells, horizontal cells and amacrine cells. Next is the inner plexiform layer. This synaptic layer is formed by the axons of bipolar cells with the dendrites of ganglionic neurons and the processes of amacrine cells. In the diagrammatic representation, this is the positioning of the inner plexiform layer. Ganglionic cell layer. Ganglionic cells are large multipolar neurons with large euchromatic nuclei. These are the second order neurons of the visual pathway. Two types of neurons are there, diffuse and monosynaptic. They receive input from the bipolar cells and their axons form the optic nerve. Branches of the central vessels of the retina are present in this layer. In the diagrammatic representation of the microscopic study of retina, you are seeing the ganglionic cells here. This is the ganglionic cell. These are large multipolar neurons. The next is the stratum opticum, that is a nerve fiber layer. Bundles of unmyelinated axons of ganglionic cells form this layer. These nerve fibers run towards the optic disc and pass through the lamina fibrosa of the sclera to form the optic nerve. External to the eyeball, all these nerve fibers will become myelinated by the oligodendrocytes. Here you can see the nerve fiber layer. You can see the axons of the ganglionic cells, they are grouped together to form the optic nerve. The last layer, that is the internal limiting membrane, it is formed by the expanded basal ends of the Muller cells, that is the neuroglial cells and the basement membrane. This layer separates the retina from the vitreous body. So, about the nutrition of the retina, the inner layers are getting nutrition from the central artery of the retina and the outer layers from the capillary plexus of the choroid. Applied histology of retina. Retinopathy means any damage of the retina which is leading on to visual impairment. The term often referred as a vascular disease. The basic cause is abnormal blood flow to the retina. Retinal detachment means there is a separation between the pigmented layer of the retina and the inner neural layers of the retina. The potential intraretinal space is widened here. We have seen the microscopic anatomy of retina. Now I am going to summarize the important points. This is the microscopic appearance of retina. We can see all the layers of the retina here, starting from the pigmented epithelium, which is close to the choroid. The next layer is the layer of rods and cones, external limiting membrane, outer nuclear layer, outer plexiform layer, inner nuclear layer, inner plexiform layer, ganglionic cell layer, nerve fiber layer and the inner limiting membrane. So, after summarizing the three topics, I am going to give a simple assessment for you. 5 questions are here, multiple choice questions. You can take maximum 3 minutes for completing this, and after 3 minutes, we will have a discussion on this. So, here are the answers for the multiple choice questions. Question number 1. Bipolar cells are histological feature of cornea, retina, lens, optic nerve. The answer is retina. Bipolar cells are seen in the inner nuclear layer of the retina along with amacrine cells and horizontal cells. Question number 2. Substantia propria is a feature of cornea, sclera, lens, conjunctiva? The answer is cornea. The substantia propria forms the major bulk of the cornea, and you can see collagen fibers in this layer. Question number 3. Canal of Slum is present at limbus, blind spot, lens, optic disc. The answer is limbus, which is otherwise called as sclerocorneal junction. This canal is lined by endothelium. Question number 4. Neuroglial cells of retina are bipolar, Muller's, hemacrine, Rhodes. The answer is Muller's. These Muller cells are otherwise called as retinoglial cells. All other cells are neuronal cells which are present in the retina. Question number 5. Optic nerve starts from the axons of bipolar cells, ganglion cells, hemacrine cells, Rhodes. Ganglion cells will give rise to axons which will be continuing as optic nerve. So, we are coming to the end of our discussion. We have discussed 3 topics: microscopic anatomy of cornea, sclerocorneal junction and the retina. Thank you for listening.